Howdy, my name is Stephanie Aranda and I'll be presenting the human herpes virus 1 and 2 diseases. This is the main topic. The information to know in regards to the human herpes virus, it is that is known as the herpes simplex virus, HSV, or as the title human herpes virus, HHV. The term that I'm going to be using throughout the presentation will be the herpes simplex virus, which is the HSV. The groups classified based on a particular disease syndromes presented. This classification is used to describe each of the various herpes simplex viruses. Enveloped double-stranded DNA viruses exist in two serological types, which include the herpes simplex virus 1 and 2. The taxonomical names for these two diseases is that one of them is a human alpha herpes virus 1 and the other is a human alpha herpes virus 2. On this slide, I'll be discussing the differences between the human herpes virus 1 versus the human herpes virus 2. The human herpes virus 1 is known widely as oral herpes, cold sores, or fever blisters. This is most commonly associated with adult herpes simplex virus pneumonia. This is seen mostly in children as they get early exposure. The causes include gingivostomatitis, pharyngitis, esophagitis, keratoconjunctivitis, encephalitis. For the human herpes virus 2, it's known for its cause of genital herpes. It is associated with 80% of disseminated disease and pulmonary infections in newborns. Sexually transmitted is often presented with no symptoms. Some situations can be transmitted non-sexually, though the cases are rare. One of the cases could be a mother can pass genital herpes during vaginal childbirth. And there is a 20% crossover presented in both of these viruses. The human herpes virus 1 can be seen on the genital location, as the same as the human herpes virus 2 can be seen in the oral location. And we'll discuss how that happens in the next slides. On this slide, I'll be discussing the viral genome of the human herpes virus 1 and 2. It is known that it is double-stranded linear DNA genome. Between HSV1 and 2, there is a slight difference in the genome, which causes a 70 to 50% homology that is shared. The genome is circularized in the nucleus and is replicated in the nucleus as well. The genome organization displays densely packed transcripts. The capsid protein viruses is distinct from most animal viruses. On this slide, I'll be discussing the genome replication. The genome replication for this viruses have a short replicative cycle. The herpes human virus 1 and 2 replication cycle takes approximately 4 to 15 hours. Like many virus replication cycles, the herpes simplex virus infection results with cell death. The homology sequence between the herpes simplex virus 1 and 2 is approximately 50%. The transcription and genome replication are nuclear, as mentioned on the last slide, which is defined as everything happening in the nucleus. After infection, the genome becomes circular rather than linear. On this slide, we'll be discussing the herpes virus replication cycle. As seen on this diagram, we all know that the herpes virus is composed of the nucleic capsid along with its enveloped proteins. There are 9 steps in total for the replication cycle allow its replication of its virions. Step 1 is attachment. Step 2, fusion, which allows the enveloped proteins to translocate the capsid into the cell membrane to the nucleus. And once it is transported to the nucleus, it releases the viral DNA into the nucleus, which allows the genome transcription and replication. Once it is over, we see the viral MNR translation happen between the nucleus and cytoplasm. And once that translation is over and done, the capsid proteins are transported into the nucleus. Once transported back to the nucleus, we see the capsids forming around the genomes. And then its final envelopment of new virus particles is transferred out to the nucleus and allows for viral egress, which then releases its virions outside the whole cell to allow for maturation. For disease transmission, the herpes simplex virus 1 and 2 will be discussed on this slide. The blue arrows indicate the herpes simplex virus 1's common transmission routes, and the red arrows represent the herpes simplex virus 2 routes. The herpes simplex virus 1 is often transmitted through oral secretions or sores from skin, so this means kissing or skin contact. It could be spread by kissing or sharing an object or etc. The common possible routes for the herpes simplex viruses 1 and 2 are oral to oral, oral to genital, or genital to genital. For disease manifestation, the congenital infection includes the skin lesions, scars, choroiditinitis, microcephaly, hydranencephaly, and microphthalmia. They tend to be presented at a localized region to the skin, eye, and mouth, or the central nervous system, or as a disseminated infection. The common clinical manifestations seen in adults are the vascular instability, hepatomegaly, jaundice, bleeding, and respiratory dysfunction. For the diagnosis of the human herpes virus 1 and 2 could be done through various tests on the patient. Two of the exams that could include are cell culture and immunofluorescence test. The cell culture, you would have to know for the ballooning of the cells, as seen on the bottom left area, which shows a picture with two arrows that display the ballooning of cells. For the immunofluorescence test for the herpes simplex virus is shown on the top right corner. As seen, there are two differentiations between the herpes simplex virus 1 and 2. This is tested for the herpes simplex virus antigen within the epithelial cell. For the herpes simplex virus 1, could see it's at a localized region within an epithelial cell. For the herpes simplex virus 2, is seen a localized region that infects multiple cells. For my sources, I have presented them in an APA format. I used a variety of sources, but my primary source that I used was from Susan Payne's book that we all have read as a class, from chapter 34, The Family Herpes Viridae. And this concludes my presentation. Now we all should understand what human herpes virus 1 and 2 are and how it affects us. Thank you.